In the last video, we talked about protein structure and how proteins are folded, but we also talked about, prior to that, how proteins are structured at the amino acid level. We talked about the amino acid being composed of three parts: the carboxyl group, the amino group, and the R group. We also talked about how the R group is the single most important component to an amino acid. When we talk about the specific or the specificity of the amino acid, we know that the R group is the only one that's different. And it is the interactions among the R group and the interactions among amino acids, both adjacent and non-adjacent amino acids, in order to determine what the sequence of that protein will be, and then ultimately how that protein folds itself to produce the function that that protein is supposed to have. In this video, we're going to talk about specific protein uses. We're also going to talk about the specific examples the IB has deemed important, or the IB has identified as the ones that we need to focus on in order for IB exams. And so we're going to talk a little bit about specific protein uses and what happens to the proteins when homeostasis isn't maintained. What happens to the proteins when pH varies from normal? What happens to proteins when temperature deviates from normal? What happens to pH when optimum conditions are not maintained? And that's ultimately denaturation, which we're going to talk about shortly. Okay, so we talked a little bit in the last video about fibrous versus globular proteins, but I wanted to give you kind of a graphic in order to kind of compile all of our thoughts. Fibrous proteins are proteins that resemble fibers, and globular proteins are proteins that resemble a glob, if you can think of it that way. Globular proteins are going to be typically rounded, and they're going to be kind of this quaternary mass of proteins. You can see in this diagram, or in this protein, there is a single kind of pink protein, there's a single yellow, there's a single green, and there's a single blue. All of those individual tertiary proteins come together to produce a fully developed, fully articulated globular quaternary protein. And these proteins are going to be very functional in their specific role. And by functional, I mean they're going to catalyze reactions. They're going to act as enzymes. Typically, enzymes are going to be this globular shape, and we know that enzymes catalyze biological reactions. These are also going to be enzymes that are important in transporting molecules and materials. Specifically, or as an example, hemoglobin is a globular protein that is also an enzyme. It is responsible for transmitting or transporting oxygen and carbon dioxide to and from the cell in order to maintain proper concentrations of those, in order to maintain cell respiration, which is what we're going to find out, an aerobic process. Okay, these globular proteins are rounded or spherical in shape. They're also mostly or generally soluble in water, and if you remember or recall from last lecture, we know that they are soluble in water because although they have hydrophobic regions, all proteins have hydrophobic and hydrophilic regions. The fact that they are globular in nature or in shape allows them to hide their hydrophobic regions inside or internal, and allows them to expose the hydrophilic regions to the outside. Okay, and so they're soluble in water, and that is directly because hydrophobic stays on the inside and hydrophilic stays on the outside. And because they are so tightly packed and so tightly coiled, they are able to keep their hydrophobic regions hidden from the water molecules that are free in solution or free in the environment that the enzyme or this globular protein is bathed in. Okay. If we were to contrast that with a fibrous protein, the fact that they are long, narrow, skinny does not allow them to adequately hide or adequately maintain a separation between their hydrophobic regions and the outside. And so if the hydrophobic region was here, let's say, that hydrophobic region is going to be exposed to water, and so therefore not soluble. It's not going to be able to dissolve correctly. It's not going to be able to hide itself because the entire protein or the entire length of the protein is exposed. If that same protein was folded into this globular rounded shape, this section, which is hydrophobic, could be the section that nestles itself into the center and disallows the water molecules that are in the solution that that enzyme or that globular protein is bathed in from contacting that section. And so, again, this is generally soluble while these are not. Okay. If we go back and forth, I guess I was kind of getting ahead of myself talking about just globular proteins, but we know the shape is obviously different. So these are a set of descriptors, a set of properties, if you will, that we are going to use to kind of compare and contrast these different protein shapes or these different protein structures, both fibrous and globular, are kind of the two divisions, the two umbrella terms we're going to classify all of these next several examples under. Okay, and so the shape of a fibrous protein, just like this one indicates, is long, narrow, skinny. It is a fiber. Okay, that's what the kind of root word of fibrous protein is. It is a fiber, and because it is long, narrow, it's going to have a lot of tensile strength. It's going to have a lot of support capabilities. These are going to be the beams and the cross members and the posts and the columns, if you will, for different structures in and around the cells. We're going to talk about those structures in a minute. But the role of a fibrous protein is structural in nature, while the role of a globular protein is functional. Remember, these are the enzymes that are going to catalyze biological reactions. The solubility, just like I said, is going to be generally insoluble in the role of a fibrous protein and soluble in the role of a globular protein. Okay, again, because they hide the hydrophobic interactions, they hide the hydrophobic zones, or the hydrophobic sections of that protein in the center, whereas these proteins, that hydrophobic zone or hydrophobic interaction, hydrophobic region, is going to always be exposed because the entire length of the protein is exposed. The sequence of a fibrous protein is very repetitive amino acid wise. Okay, we are going to just repeat amino acids one after another, after another, after another, after another, all the way through the length of a fibrous protein. Now, these are going to be repetitive amino acid sequence too, but they are more irregular because there are more complex disulfide bridges, disulfide bonds, salt bridges, stuff like that, that are going to impact the structure ultimately and then impact the function of that protein. Stability, these are less sensitive and so therefore more stable. So I'm going to put kind of an up arrow. These are more stable. They are less sensitive to the changes of heat, pH, salt, water concentration, stuff like that. Things that are going to deviate from the norm, these remain more stable. It makes sense. If they are more structurally sound and they are more produced for structural integrity, then it makes sense that they would be more stable in an unstable environment or in a changing environment. Globular proteins are more sensitive and so therefore less stable and therefore easier to change because there are more hydrogen bonds. Remember, hydrogen bonds are weak, and the more hydrogen bonds we have in a particular molecule, the less stable it's going to be when we start talking about adding heat, pH, salinity, stuff like that. Okay, if we deviate from the normal, these are going to remain less stable. They're going to be more sensitive to changes. They're going to be more sensitive to denaturation. Okay, these typically are going to denature faster. They're going to denature more intensely, and these are going to remain more stable. Okay, examples of fibrous proteins are all of these. Now, all of these are not going to necessarily be needed for IB exams, but I'd like to go through and kind of give you an idea of what these are. Collagen and elastin, the two kind of bookends of this, are related to skin strength, skin function. Okay, collagen and elastin give the structural strength to skin. These are going to obviously break down as we age, which is why we wrinkle as we age. But collagen and elastin are the main fibrous protein component of skin. Myosin is a structural component to muscles. Fibrin is the structural component or the component to blood that allows it to clot. Okay, so when you think of like scab, you think of blood clotting, it takes a certain structural protein that allows that clotting to work. Okay, this is kind of the lattice that is going to allow the platelets and things to stick, give it structural support, form the scab, and then eventually produce the healing properties that that scab will allow you to do. Actin is one of the main components to a lot of cytoskeleton structures. Okay, actin is going to enable cells to support themselves internally. Cytoskeleton is a main structural support system to cells. Actin also is going to help maintain cilia, microtubule, doublets, or it's also going to allow for cell division to take place. Okay, keratin is the main component to hair, or the protein component to hair and feathers. So we have all of these different examples, from our skin to our hair to our cell cytoskeleton support system to muscle fibers, and also the ability for blood to clot. On the globular protein side, we have several examples as well. I've already kind of outlined what hemoglobin is. Hemoglobin, remember, is the main enzyme in blood that allows for O2 and CO2 to be transported to and from the cell. Insulin is a globular protein. It is a hormone. It signals the absorption, or for the absorption, of blood glucose. Immunoglobulin is part of your immune system. Okay, we're going to talk about it in more detail. It basically makes up the antigens, which are going to, or it makes up the antibodies, I should say. Antibodies are going to have specific antibody-antigen interactions. That gives the immune system the ability to identify and attack foreign and possibly invading cells. And then catalase is an incredibly common enzyme, and it is found generally in the liver of animals, including humans, and that allows for the breakdown of hydrogen peroxide. Now, I know hydrogen peroxide, you probably think that's in my bathroom cabinet. Why do we have enzymes that break that down? Well, hydrogen peroxide can be a product of cellular respiration and a lot of the metabolism and cellular energetic processes that take place in the cell. And in order for our cells to combat and protect ourselves against oxidative stress and oxidative free radicals in the nature, catalase needs to be able to detoxify. It needs to be able to kind of protect ourselves against oxidative damage. Okay, so these are several examples. We have both structural fibrous proteins and globular functional proteins, and so we want to kind of outline a few of those and their particular roles, or specific roles in the body. Here's just another type of table that outlines even more varieties. Notice that some of these examples are red. Okay, we have Rubisco, collagen, insulin, rhodopsin, immunoglobulins. These in red are all examples that the IB has deemed important for you to focus on as we work through the very varied and vast functions that proteins allow us or afford us to have. Okay, we talked about enzymes like Rubisco being catalysts. Catalysts are the molecules in our cells or in any chemical reaction which are going to do two things. They're going to speed up the rate of reaction, and they're going to decrease the amount of activation energy required to actually activate that reaction. Cytoskeleton, just like tubulin and actin, they're going to give structural support to animal cells. They're going to give the cells their shape. They're going to allow the cells to move. They're going to allow chromosomes to be moved around, and they're going to allow the cell to divide during cell division, which is called mitosis. Okay, spindle fibers are not necessarily cytoskeleton per se because they kind of come and go, but spindle fibers are definitely linked to protein because they are produced by or made from tubulin and actin. Muscle contraction I talked about before. Myosin is paired with actin. Myosin and actin are going to interact to allow muscles to contract and relax. That gives vertebrates, it gives animals the ability to move and lift and climb and run and swim. Really important when we talk about life as we know it. Fibrous proteins, remember, give a lot of tensile strength. Okay, that's basically the strength that allows a load to be put on a fiber as you pull, okay, or as you stretch it. Skin, tendons, ligaments, and blood vessel walls allow for a lot of tension. They are very stressed. They are definitely maintained tight or kept tight, and that allows for structural support to ligaments or structural support to joints, I should say. Collagen, remember, is one of those examples we highlighted on the first slide, and collagen is definitely found in skin. Okay, blood clotting, proteins found in the blood are clotting factors or act as clotting factors, which allows blood to coagulate. The reason blood coagulates when it is exposed to air is because the proteins that give the blood its structural support start drying or start coagulating, start forming a net or lattice structure, and that allows for more things to attach itself and eventually forming the scab. Okay, that is definitely moderated or mediated by protein. Transport. Proteins have the ability to transport materials. There are proteins in blood like hemoglobin, talked about before, that are going to transmit oxygen, carbon dioxide, iron, lipid all the way throughout the blood. Going to transmit that to and from the lungs, to and from the brain, to and from the heart. Hemoglobin is the main transporter in blood for oxygen and carbon dioxide. Cell adhesion. There are membrane proteins, specifically glycoproteins, that allow cells to stick to each other and to stick to other types of materials, which give us the ability to produce tissues. Okay, the fact that our heart cells stick together and form a heart gives us the ability to have heart tissue, and the fact that we have lung cells that stick together using these cell adhesion proteins allows us to have lung tissue. Membrane transport, we call these channel proteins. Okay, channel proteins are really, really important when we talk about facilitated diffusion, active transport, electron transporting for cell respiration and photosynthesis. All of these channel proteins are going to be specific in a lot of cases for the materials that are allowed to transmit through them or that are allowed to transport through them. Proteins that act as signals in the body, like I said in the first slide, insulin is a hormone that is going to signal the body to absorb blood glucose. Receptors, proteins can act as receptors. These are binding site for hormones, neurotransmitter stimuli. These give you the ability to perceive stimuli. Rhodopsin is a specific protein in your eye that allows you to see and absorb light, which allows you to see. DNA packaging, histones. Histones are proteins that are going to be in charge of or acting as spools that we're going to supercoil or condense our DNA around. So chromosomes in eukaryotes, remember, are long stringy strands of DNA. Those long stringy strands of DNA are going to actually wrap themselves around little proteins, and those proteins specifically are called histones. So those histones are going to be in charge of supercoiling or condensing DNA. That allows for more DNA to fit into a smaller area. Proteins play a big role in immunity. Remember, antibodies, like I said in the previous slide, immunoglobulins. Antibodies are going to be produced by our immune system to detect and adhere to antigens. Antigens are specific surface markers that are on the surface of everything. Okay, our individual cells have antigens. If this is a cell, an antigen could be as simple as a marker that has that particular shape. And an immunoglobulin is going to have a receptor that is going to have the same shape. This antibody is going to interact and adhere itself to this antigen, which is going to allow our immune system to identify this cell as either foreign or safe, right? It's either our cell or it's a foreign cell. If it's a foreign cell, we want to get rid of it because it's probably a bacteria, a virus, something like that. Okay, it's something we don't want to have. These antibody-antigen interactions is the reason why you can't just give any blood to anybody, right? We have blood types. We have AB, and O blood types. Those are directly talking about the antigen-antibody interactions. It is specifically, if you're type A, you have the type A antigen on your red blood cells. If this is an RBC, red blood cell, and it has the A antigen, then you are type A. You cannot give type B blood to yourself, or you cannot receive type B blood because your antibodies would basically identify that as foreign and would attack it. That is bad. Okay, so let's go through some of these examples. Remember, Rubisco is one of those that was identified as the IB as important. And so what is Rubisco? Rubisco is kind of a weird name, but it is really just a combination of the full name of that particular enzyme. Rubisco, Ru, bis, and co. Co is carboxylase, bis is bisphosphate, Ru is ribulose. So ribulose bisphosphate carboxylase is the entire name of that particular enzyme. You can see it sometimes written like this, where it is capitalized for the first letter of this full name. Rubisco, or ribulose bisphosphate carboxylase, is the enzyme that catalyzes carbon fixation. So if you look at this diagram, you'll probably recognize it as being part of the photosynthetic cycle. Okay, in fact, this is the Calvin cycle. Okay, and during the Calvin cycle, carbon dioxide is fixed, or carbon fixation happens. Carbon dioxide, which is located in the atmosphere, is brought in by Rubisco and is actually added to the 3-phosphoglycerate. This is an intermediate molecule within this particular pathway. Now, the purpose of this lecture is not to understand photosynthesis or this process specifically. We're going to get to that in a later video. The purpose of this video is to talk about proteins and specifically Rubisco. I just wanted to outline Rubisco's role in photosynthesis as a whole, and so that we understand exactly what this particular protein does and the importance of that protein in producing energy for the producer level, but then also producing energy for the consumer level once these particular plants, or once the plants that are producing the glucose down here get by consuming them. Okay, and so this is one of the most abundant proteins on Earth. That's because the base of the food chain are the plants. Plants make up the most biomass, or the biggest level in terms of biomass. Requires a lot of biomass in order to feed all of the consumers found on the planet. All of the producers that produce glucose by utilizing carbon fixation are going to need and implement this particular enzyme in order to do that process. This enzyme is found in very high concentrations in leaf and algal cells. These are the sites of photosynthesis. Okay, so this is where photosynthesis takes place. It makes sense that the enzyme that's responsible for fixing carbon is found in the cells or in the leaf tissue where photosynthesis is going to take place. That's where gas exchange happens. This is where the enzyme is required in order to function correctly. The enzyme is very inefficient, however, and requires high concentrations in order to function correctly. The leaf tissue has to maintain very high concentrations of this Rubisco, or ribulose bisphosphate carboxylase, in order to function correctly, and in order to keep up with the demands, the glucose demands placed on that particular cell. So next we have collagen. Collagen is a specific protein found in the human body, specifically found in skin. So you'll notice that collagen is a very abundant protein. Okay, in fact, it is the most abundant protein found in the human body. Skin is a substantially large, or an incredibly large organ in the human body. We have a lot of skin tissue. Skin tissue is very important in maintaining boundary between inside and outside of our body. It's kind of our first line of defense within our immune response. And collagen is the most abundant protein within that particular tissue. Okay, so collagen, let's just kind of look at it, break it apart, look at the specific fibrils and fibers and the makeup of that particular fiber. Collagen is made up of 3 polypeptides that are wound together. So this would be a structural fibrous polypeptide, this would be a long polypeptide, and then this one would be a polypeptide. We have 3 individual fibrous polypeptides that are wound together, kind of like a rope would be wound. Inside of these fibrils, which are larger bundles of these particular polypeptide triplets, these triplets are linked into a fibril, and it's a group of those particular polypeptide triplets. Well, then these groups are grouped within fibers that make up the collagen, or make up the structure that our collagen is packaged in. Okay, so collagen, you can see, make up this lattice structure, and notice that as we age, the collagen gets less and less concentrated. It starts breaking down, it gets weaker and weaker, and because our skin doesn't have as strong of a support system to hold this particular roof up, if we use a building analogy, if a building has adequate structure, the roof stays very tight, taut, it is held up adequately. But as we start breaking down the infrastructure that holds our roof up, it's going to start sagging and it's going to start collapsing. And that's exactly what happens to our skin when the support system, or the structural support system, starts degrading. Okay? Collagen is made up of, again, a protein, and is the structural protein that creates the strength associated with all of our tendons, bones, teeth, skin, blood vessel walls, etc. All of those things that require a high level of structural support, structural integrity. Collagen is going to be the protein that does that. Next is a hormone called insulin. Insulin is a hormone that is going to regulate our blood glucose levels, and so when we eat a meal, it's going to just kind of put a flow chart down here. As we consume food, our glucose level is going to increase, okay, because we break down our food into individual monosaccharides, that is glucose. It's going to flood to the blood, and our blood glucose level is going to spike. In response, as a feedback mechanism, in response to our glucose levels increasing, our body is going to release a hormone called insulin in order to bring blood glucose levels down. Okay, what happens if our insulin, or what happens if our blood glucose levels stay high? If blood glucose stays high, it is going to produce a dangerous condition called ketoacidosis, which is going to basically mean that we're going to have high levels of ketones in our body, in our bloodstream, because glucose can't be utilized properly. We cannot regulate the glucose levels correctly, and because we can't regulate those glucose levels correctly, we're going to get a condition called ketoacidosis, which is only a condition when the hormone insulin is not implemented correctly. Okay, normally ketoacidosis is not a dangerous concern, or not a big concern or a big threat, because we typically have insulin levels that are going to naturally increase or decrease the blood glucose levels and stored glycogen levels to moderate and maintain homeostasis as we kind of eat and then fast, eat fast, eat fast, right? We're not consistently grazing like a lot of animals would, and so we have kind of blood sugar spikes and dips, and it is hormones like insulin that is responsible for maintaining homeostasis and maintaining kind of an even level of blood glucose in order to maintain homeostasis. Okay? Insulin, again, is a hormone that is a type of protein, and normally insulin is at high enough levels in the body that is going to accurately maintain glucose levels in the blood, but if you are diabetic, or if diabetes is acquired, specifically type 1 diabetes, that is the type of diabetes that is going to that happens when the person can no longer produce adequate levels of insulin. So it's kind of a delayed onset typically. But those types of disorders, or type 1 diabetes, is going to require periodic injections of insulin in order to avoid this ketoacidosis and maintain proper blood glucose levels. Next protein is rhodopsin. Rhodopsin is a specific pigment that is located in the disc membrane region of a photoreceptor cell. These are located in the retina, which is the portion of the eye, or the portion of the eye that's in the back. And the retina is the portion of the eye where all of the light that comes through the cornea and the pupil, I should say, focuses on in order to be in order to be kind of changed into an impulse or a signal that is traveling to the brain in order to allow you to perceive the stimuli that you're perceiving. And these rods and cones that are cells that have very specific structures that allow them to function in a very specific way. Again, we're going to talk about all of this later in the syllabus, but they contain a pigment, and just like your skin contains a pigment to help protect the skin cells from damaging effects of UV radiation, these specific cells, while they don't have to protect themselves against UV radiation, because typically you don't have large amounts of radiation kind of entering the retina portion of your eye, you do need to have pigments that allow this particular cell to absorb light, because without the absorption of light, you really can't perceive signal or stimuli. You really can't perceive the specific color frequencies. It's not going to allow you to see the right color. It's not going to allow you to change those light frequencies into a chemical impulse that is sent to the brain that allows you to perceive the stimuli. Okay, this is a pigment. The pigment is a protein. Like I said, it's located in the retina portion of the eye, and because it allows you to perceive or absorb the light, it's going to allow you to see in very dim light situations. It's very good at absorbing light, and it is going to change its conformation, which means changes its shape, and it's going to mediate a signal sent to the brain, okay, in the form of a nerve impulse. And that nerve impulse, that signal that is sent from the eye through the optic nerve to the brain, is going to allow you to perceive the stimuli and create an image in your brain, which is going to allow you to kind of figure out a way to manage the situation. It's going to allow you to not only perceive the stimuli, but allow you to kind of reflect on and make a decision based on that stimuli. Next protein is immunoglobulins. Immunoglobulins are a form of globular protein that are part of the immune system, and this is kind of an actual image, or what the actual protein would resemble. Notice the Y shape. This image is kind of a more artistic rendition of the particular protein, but it allows us to more discreetly identify the antigen binding site, which, notice, based on the different antigen shapes, has a specific shape that will fit a specific antigen. So antibody and antigen interactions are the key to immune response. Antibodies are the proteins. Antigens are proteins that are on the surface of a cell. Let's say that this is a bacteria cell, and on that bacteria cell is an antigen that is identified by our antibody based on this interaction, okay? Because this is the same shape, it's going to create a bond. It's going to fit into this particular antigen binding site, and because it fits and interacts together, this particular antibody is going to tell our body in the form of an immune response to not only kill or destroy that particular bacteria cell, but is also going to ramp up our defenses, okay, increase defense by producing more of these antibodies, so that if we are exposed to higher concentrations, or if this particular bacteria replicates and produces more, we are well equipped, or more well equipped, to handle the situation, so that we don't succumb to the side effects or the drawbacks of a bacterial infection. Okay, so the antibody is going to recognize the unique molecule of the pathogen that is called an antigen. They look like this. All antigens are specific to, or I should say most antigens are specific to the antibody they are going to interact with. Notice that this particular shape is not going to fit well in this antigen binding site, and this antigen binding site is not going to work very well at recognizing this antigen shape. This particular antigen is going to be the one and the one only, or the only one that fits into this antigen binding site. It is antigen antibody specificity, just like enzyme substrate specificity, specific antibody, specific antigen, specific interaction, specific immune response. Okay, each tip of the Y remember is the antibody. Each tip of the Y contains a paratope that is specific to one particular antigen. I've already talked about that, right? They bind together, and because they bind together, they are going to create an immune response. Okay, the binding of these two is going to create a response which your body will recognize as either identifying something that is foreign or something that is common to your body, something that should be there versus something that shouldn't be there. There is great variation among these antibodies because there are great variations among the antigens associated with bacteria and viruses and parasites and cancer cells and stuff of that nature. Okay, there's a huge range of pathogens, which means there's a huge range of antigens. We need to have a huge range of antibodies in order to interact and recognize all of those antigens. Last protein, I believe, is spider silk. And while spider silk is something that we can actually see with the naked eye, which is unlike the other proteins that you can't see with the naked eye, this is a very large collection of protein fibers that are spun by arachnids in the form of webs. Okay, and so not all spiders produce spider webs in the sense that we know of them, like this one is showing. Some spiders are going to create webs that are more tunnels in the ground, but all spiders can produce spider silk, and all spiders, while not spinning webs per se, will protect offspring in the form of nests or wrap up prey or suspend themselves, move themselves, transport themselves with the use of silk. Okay, all spiders produce silk. The silk is a particular type of protein that is very structurally sound. It is very supportive in terms of being able to collect prey items, hold prey items, support the weight of the spider, and there are actually 7 different types of silk. We're not going to go through all of the different types because that's not really a requirement at this point. But spider silk in general has very, very good mechanical properties, and it exhibits a very high tensile strength and extensibility, which means it can support a lot of weight and is very, very hard to break. Okay, large amounts of energy before breaking, and dragline silk, which is the type of spider silk that actually can suspend a spider and move a spider, is very, very comparable to high-grade alloy steel. So the fact that we can move very, very high or very, very heavy materials with the use of a crane, let's say, with high-grade alloy steel cabling, spiders could do the same exact thing with their particular dragline silk. So very awesome form of protein. It has a very good function. It has also a very good structure that allows it to function that way. I can't stand spiders, hate spiders. I don't even like seeing this because it kind of makes me freaked out. But spiders are very interesting critters regardless. So, out of the types of examples and into the specific proteome, before we enter into denaturation talk, which is how we're going to end it. Proteome is the entire set of proteins that can be or that is expressed by a genome of a particular organism. Okay, genome, remember, is the entire gene or the entire list or set of genes exhibited by or expressed by a particular organism, cell, tissue. And so as complexity increases in terms of organism complexity, the amount of proteins increases. So human proteome consists of about 92,000 proteins, which are made from about 19 to 20,000 protein-coding genes. What does that mean? That means that multiple genes can produce, well, it means that one gene can produce multiple proteins. How does it do that? It depends on which part of the gene is turned on and off. So as we turn, and I know we're not really into genetics yet, but if you look at like splicing, if we edit or turn on and off certain aspects of genes, you can produce a different protein. And that has to do with really the demands placed on the cell. If the cell needs a specific protein, it's going to turn on that specific part of the gene in order to make that particular part of the protein. If it doesn't need that particular protein because of dietary reasons or structural reasons, then it's going to turn that particular protein off. The cell wants to be efficient. The cell does not want to waste energy. It does not want to stockpile a lot of enzymes or a lot of proteins that it doesn't need. And so it's going to turn on and off specific sections or specific genes in order to turn off specific protein production, and is going to turn on the specific protein production when it's needed. Okay, more than one protein can be produced from one gene, and it has to do with splicing. We'll get into that later. What happens though when proteins are not kept at optimum temperatures, like when heat is applied to a protein? It's called denaturation. If this is the structural shape of a protein in normal conditions, okay, normal conditions, the protein should look like this. If we increase the heat, we are going to cause all of the hydrogen bonds, which I'm just going to denote by these dotted lines. If these dotted lines indicate hydrogen bonds, and these hydrogen bonds are the interactions that are holding this particular protein intact and in its conformation or its shape, then the heat, as it increases, is going to start breaking these hydrogen bonds. And as we break these hydrogen bonds, the structural integrity of this protein is going to start unfolding. The hydrogen bonds are not going to hold it together in this three-dimensional shape. And as the hydrogen bonds and the disulfide bridges and the salt gaps and the salt bridges start breaking, it's going to unravel this protein. And as the protein unravels, it's not going to function correctly. And so this is an example of denaturation and can have serious, life-threatening, fatal consequences if this is a vital protein or a vital enzyme in terms of cellular metabolism. Okay, so this would be, just say abnormal. Why is it so bad to have a fever that exceeds 105? Or why is it bad if your core body temperature increases a few degrees? Why is it bad for your core body temperature to drop a few degrees? Hypothermia and hyperthermia are serious health concerns, because what happens when your enzymes become unstable? What happens when the enzymes denature? They don't adequately hold on to the proper substrate. They don't have their proper substrate enzyme specificity. All of your enzymes start looking like this, and that has serious consequences for the survivability of the cell and the survivability of the organism. So as the temperature increases, the hydrogen bonds are going to be disrupted. The nonpolar hydrophobic and hydrophilic interactions are going to be disrupted. The shape of the protein is going to be pulled apart. Alcohol in a solution is going to disrupt hydrogen bonding as well. Acids and bases, if we alter pH, if we increase pH and get more alkaline, or if we decrease pH and we get more acidic, it's going to disrupt the salt bridges. It's going to dissolve those or break the salt bridge interactions, and it's going to cause these proteins to pull apart. Again, as those proteins pull apart, it's called denaturation. That has serious consequences for the biological reactions that have to be catalyzed. The biological reactions cannot be catalyzed anymore, and therefore requires more energy than the cell is willing to or is unable to utilize, okay, or spend. Denaturation reactions are not strong enough to break the peptide bonds, so you'll never see it start decomposing or breaking the peptide bonds that are holding this chain of peptides together, which is why we didn't actually lose any of the material the protein was made by. We are just losing the conformational shape of the protein because we are breaking the weak hydrogen bonds, the disulfide bonds, and the salt gap or the salt bridges. Okay, all of those weak things, weak bonds, are going to decrease. They're going to disrupt or be disrupted, and what is the long-term implications of this? Denatured proteins are not normally going to return to the original or permanent state. They are going to become insoluble if they were soluble, and they are going to form a precipitate, which is typically going to fall to the bottom of the solution that protein is in. And so if that protein is in your body and it becomes denatured, it's just going to precipitate out of that solution and become useless. It is not going to be switched back. Once it looks like. This, it cannot go back and reconform itself or reshape itself. It's going to be broken or restructured forever. Okay? I think that's where we're going to end it. That is a relatively long lecture on protein structure and the specific examples that IB has deemed important. Bring your questions to class. I'll see you later. Bye.